Hello. Very good evening to the student who have joined. Can you please let me know if my voice is audible to you? Please let me know in the chat box if my voice is audible, and also please let me know if you can see the presentation clearly. Let me know if everything is all right with the audio and also with the presentation. Okay, thank you for confirming. We will be starting at 6 p.m. Till then, kindly wait. Let more students join, then we will start today's session. Please wait till it's 6 p.m. Then we will start today's session. So, very good evening to the students who have joined. We will be starting our session at 6 p.m. So, kindly wait. Let's wait for 3 to 4 minutes and then we will start today's session. Okay. I hope my voice is audible to you all and I hope the presentation or my screen is visible. If not, please let me know through the chat box. Okay. Very good evening. Let us wait 2 to 3 minutes. So, I think I have already shown you where to find the lecture videos and presentation. I hope you all are able to now access the lecture materials. So you have to go to the SWAYAM portal. Again, let me show you before starting the session. You have to go to the SWAYAM portal and then you have to, on the left panel, you have to click on the problem solving session 2026. If you click on that place, you will be redirected to a page where you will be finding the class recording as well as you will be finding the presentation. The presentation will be inside a folder which looks like this. There you will be finding each and every week's course material. Okay. After each session, I upload all the materials that night. So you can check after some time or the next day, you will be finding the lecture video and presentation. Let's wait for some time and then let us start today's session. Okay, so it's almost 6 p.m. So let me start the session by introducing myself. Yep. So, very good evening to the students who have joined. My name is Mousumi Deb and I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL SWAYAM course of Medicinal Chemistry and this is our week 2 live session. Before starting the session, let me clarify a few things. There will be a problem solving session every week on Monday from 6 to 8 p.m. Okay, and the classes are on Google Meet and the link will be same. This is the link for our entire semester. Okay, you have to use this particular Google Meet link to access the Monday classes which starts from 6 to 6 p.m. So if any network disruption happen from my side, do not leave the session. I will be trying to rejoining as soon as possible and the call is open. So if you have any problem because of your network or anything, you can rejoin at any time. This during the session, you can post your doubts, your queries, your question, everything to the chat box. Since I'm recording the session and this recording will be provided to you after the session, so that I can record the session without any hassle. I have muted your microphones and cameras, but the chat box is always open and I'm monitoring the chat box every now and then. So if you have any doubt, any query, anything, that you want to say, you can let me know through the chat box. Okay. As I have mentioned, I am recording the session. So the recording and this presentation, which I am showing you today, both the things will be provided to you after the class by tonight or by tomorrow morning. Okay. So where you have to find these things, I have shown you earlier also. You have to go to the NPTEL SWAYAM portal. Okay, where is the weekly announcements are there. So you will be finding there is a left panel where there is a problem solving session January 2026. Here if you click on this problem solving session recording, you will be redirected to a page which looks like this. Here you will be finding the YouTube links, which are the class recordings, and a Google Drive folder. This Google Drive folder contains your presentations. So already we have one class which was in our last week. So the presentation has been uploaded, and this presentation which I am showing you today, it will be uploaded after the session. Okay. So what we will be doing in today's session, today it is our week 2 live session. So we will be discussing the week 2 course content of this course of medicinal chemistry. Okay. So how we have done it in our last class, that I have shown you around 20 MCQ questions. All the MCQ questions are related from the particular week's course content. Okay. And each MCQ question have 4 different options. After showing you a question, I will wait for some time, and then I will be checking your answers via chat box. You have to let me know in the chat box A, B, C or D which of the following will be the correct answer for a particular question. Okay. After checking your answers, I will show you the particular question, the particular correct answer, and we will discuss that particular topic in very elaborate manner. Okay. So that if you have asked any kind of descriptive questions in that particular topic, you can answer that easily. Okay. So let us start with, let us see first what is the course content of week 2. So the week 2 course content, it has the enzyme catalysis part in 2 parts. Okay. Then we have binding forces, protein structure and function, introduction to receptors, receptors types and functions, and we have the binding interaction, ion channels and receptors, a little bit of details. So what have we have discussed in our last class? We have discussed basics about medicinal chemistry, about drug discovery. We have discussed about amino acids, protein, acid, base and these things. Okay. So today now we will be moving toward the an important topic of the medicinal chemistry, which is the enzyme catalysis. Okay. So let us see what is the first question of today's session. So the first question of today's session is, which one of the following statement regarding the enzyme is not true? Your options are: number A, enzymes are one kind of proteins. Number B, they can change the rate of the reaction. Number C, they get consumed in the course of reaction. And number D, they are site specific in nature. Which of the following statement regarding the enzyme is not true? Read all the options very carefully and then let me know. See, whenever there is a question like true, false, or correct, incorrect, read the options very carefully. Okay. I will wait for a minute, then I will check the chat box. Let me know which of the following statement regarding the enzyme is not true. What do you think? Number A, enzymes are one kind of proteins. Number B, they can change the rate of the reaction. Number C, they get consumed in the course of the reaction. Number D, they are site specific in nature. Whatever you think is correct answer for problem number 1, you can let me know in the chat box. For answering to in the class is not, there is no marking. So there is, if your answer is wrong, there won't be any kind of negative mark or so. But answering is very important. Otherwise, I won't understand that you are following or not. Okay. Which one of the following statement regarding enzyme is not true? Number A, enzymes are one kind of proteins. Number B, they can change the rate of the reaction. Number C, they get consumed in the course of the reaction. And number D, they are site specific in nature. Okay, let me check the chat box. Let me check if there is any answers for our problem number 1. Okay. Okay, I have got many answers and everybody thinks that number C is the correct answer for problem number 1. Okay, let me check if number C is the correct answer or not. It is good that all of you are answering. So it is, let me check. So the correct answer, the statement which is not true will be, Yes, it is number C, that they get consumed in the course of the reaction. This statement is not true. So what is the enzyme and what is the process of the enzyme catalysis? So enzymes are basically, you can say it is a kind of catalyst which catalyzes the reactions of our body. Okay, so this is the rate graph that demonstrates the activation energy, stabilization by the enzyme. So this is the starting material and this is the energy of the protein. We know the energy of the protein, the product of the should be less than the starting material to get stabilized. Okay, so this is called the transition state. The starting material have to acquire this amount of energy, which is known as the activation energy, to go to the transition state. Then it finally creates the product. Okay, so this is the normal reaction, I mean graph without any enzyme. Now with enzyme, what will happen? As you can see, the enzyme goes to the in presence of enzyme, the particular starting material goes to another pathway to have a new transition state which has lesser energy than that of the that one without the enzyme. Okay, as you can see, this blue one is the new transition state which is with enzyme, and the black one which is without enzyme, it is higher of the energy. So as you can see, the enzyme is decreasing the activation energy for attaining a new transition state, and the product will remain the same. This is the process of the enzyme catalysis. So enzymes which are a protein, it provides a surface. So if the surface, this is known as the active site. In the active site, the substrate can bind like this, and we have something which is known as the enzyme substrate complex. Then reaction happens and the enzyme, it creates the product. Okay, and the product creation, after the creation of the product, the product is released from the active site. So enzymes provide the reaction surface and the suitable environment. Enzyme bring the reactants together and position them correctly so that they easily attain their transition state configurations. Enzymes weaken bonds in the reactants. Enzymes may participate in the reaction mechanism. Enzymes form stronger interaction with the transition state than the substrate or the product. So these are some specific comments regarding the enzymes, how they work and everything. Okay, and this is the two different theories of the enzyme which we will discuss in our upcoming slides. So one is the lock and key theory, which shows that the substrate and the active site of the enzyme, which are exactly same in the size and shape, and the enzyme in the active site, the substrate fits perfectly like a key to a particular lock. So this is the lock and key theory. And another theory we have, which is called the enzyme induced fit hypothesis, where it can be seen that the enzyme does not have any perfect shape for the substrate, but in presence of the substrate, it modifies itself, and so that the substrate can bind. So these are two different theories which we will be discussing in our upcoming slides. Okay, so yes, number C was the correct answer for problem number 1, and you have answered it very clearly, so very good. Now let me go to the next slide. Problem number 2, it says, which statement is false? Your options are: number A, induced fit hypothesis is more accepted than the lock and key theory. Number B, the most accepted model for enzyme substrate complex is the lock and key theory. Number C, the enzymes and substrates are rigid according to the lock and key theory. And number D, induced fit hypothesis explains why some enzymes can bind with a range of the substrate. Let me know A, B, C or D, which of the following statement is false. Again, let me repeat. Number A, induced fit hypothesis is more accepted than the lock and key theory. Number B, most accepted model for the enzyme substrate complex is the lock and key theory. Number C, the enzymes and substrate are rigid according to the lock and key theory. Number D, induced fit hypothesis explains why some enzymes can bind with a range of the substrate. Which of the following statement is false? That you have to identify and let me know in the chat box. Again, I will wait for a minute, then I will check the chat box, read the options carefully and let me know A, B, C or D, which of the following statement is false. Which of the following statement is false? I will wait for a minute, then I will check the chat box, okay? Number A, induced fit hypothesis is more accepted than lock and key theory. Number B, most accepted model for enzyme substrate complex is the lock and key theory. Number C, the enzymes and the substrate are rigid according to the lock and key theory. Number D, induced fit hypothesis explains why some enzymes can bind with a range of the substrates. Let me check the chat box. I hope I will find some answers there. Okay. Yeah, so I got number C, number C, number B, number B, number C, number B. So I have got number B and number C as the answers for the problem number 2. Okay, let me check which of the following is the correct answer, number B or number C, or is there any different correct answer for the problem number 2? So the correct answer, the statement which is false, it will be, it is number B. The most accepted model for enzyme substrate complex is the lock and key theory. This statement is false. Now let us understand both of the theories in detail. So, in the past, it was thought that the substrate fitted its active site similar the way a key fitting to a lock, which is Fisher's lock and key hypothesis. As you can see, I have mentioned the enzyme and substrate, they both fit with each other perfectly. Like now we have a particular key for a particular lock and the key fits on the lock in the perfect manner. The shapes are the same. So, in the lock and key theory, it is presumed that the enzyme's active site and the substrate both fit to each other perfectly. Okay. So both enzyme and substrate were seen as a rigid structure with the substrate as the key fitting perfectly to the active site of the enzyme, which was thought as a lock. However, this scenario does not explain how some enzymes can catalyze a reaction on a range of different substrate. It implies instead the enzyme has an optimum substrate that fits it perfectly, whereas all other substrate can fit less perfectly. This in turn would imply that the catalyzed reaction is only efficient for the optimum substrate. As this is not the case for many enzymes, the lock and key theory analogy must be invalid. It is now proposed that the substrate is not quite the ideal shape for the active site and that it forces the active site to change its shapes when it enters, a kind of molding process. This theory is known as the Koshland's theory of induced fit, as the substrate induces the active site to take up the ideal shape to accommodate it. It is shown in this figure. As you can see, the enzyme's active site, it does not have an ideal shape to fit the substrate properly, but in presence of substrate, when substrate is nearby, it modifies itself, it modifies its active site structure to have the substrate inside. Okay, so this is known as the induced fit hypothesis. Now let us go back to the options. The option number 1, it says that induced fit hypothesis is more accepted than the lock and key theory, which we have seen. The most accepted model is of the intercepted complex is lock and key theory. It is not, this is false, because as I have mentioned that this is not in the case that both the enzyme and active site and the substrate will be perfectly fitting with each other. The enzymes and the substrate are rigid according to lock and key theory. This is again true. And number D, induced fit hypothesis explains why some enzymes can bind with a range of substrate. This is also true. These things, it cannot be explained by the lock and key theory. Okay, that is why the answer is number B. Okay. So now how this induced fit happen? Okay, so now the question arises, how the in presence of the substrate, that particular active site of the enzyme molds itself to accommodate the substrate perfectly. So this is the one of the example of induced fit hypothesis. The substrate such as pyruvic acid, so this is the structure of pyruvic acid, this is the substrate for a particular enzyme, let us see. Okay, might interact with a specific binding region in the active site of the lactate dehydrogenase is the enzyme via one hydrogen bond, one ionic bond and one van der Waals interaction. So as you can see, this particular pyruvic acid, it can interact with the van der Waals interaction, with the ionic interaction and also with the hydrogen bonding interaction. Okay. However, if the fit is not perfect, the three binding interactions are not ideal. For example, binding groups might be slightly too far away to the corresponding binding region with the active site. In order to maximize the strength of this bond, the enzyme changes its shapes that the amino acid residues involved in the binding move closer to the substrate. So as you can see, initially when pyruvic acid enters, there is only van der Waals interaction that happens. It happens, and hydrogen bonding and ionic bonding, they are too far away. But in presence of the pyruvic acid, which is the substrate, the enzyme molds itself. As you can see, the structure of enzyme, it changes. Okay. Now from this to this, the structure of enzyme, these are the enzyme part, okay. They changes itself so that the ionic bond, the hydrogen bond, van der Waals interactions, they happen with a very good interaction intensity, okay. So to have a very good binding. This is the example of induced fit, how the enzyme molds itself around the substrate to have the better interaction in the active site, okay. That is how the induced fit happens. So is it clear? In the first two questions, do you have any doubt or any query? If you have any doubt in the first two questions, you can let me know now, or at the end of the session also you can let me know if you have any problem, okay? So the correct answer, okay. Thank you for, if you have any doubt in upcoming questions, please keep letting me know, okay? So if there is no problem, let me go to the next question. So there is another thing which I have not explained. So the theory of induced fit helps to explain why some enzyme can catalyze reaction involving a wide range of substrate. Each substrate induces the active site into a shape that is ideal for it, and as long as molding process does not distort the active site so much that the reaction mechanism proves impossible, the reaction can proceed. The range for the substrate that can accept it depends on the substrate being correct size to fit the active site and having the correct binding groups in the correct relative positions. So now let us say you are given to write a short note regarding the induced fit hypothesis. I think there is sufficient information in this slide that you can form a particular short note around for the induced fit hypothesis, okay. And always remember, whenever you are writing something, if you can give figures, if it is possible for you to draw the figures which are simple. So these figures are very simple, right? So if you can, you have to do the figures, okay. So now let me go to the next problem. Problem number 3 says what is the number of the major class of the enzymes? Major classes of the enzyme. Your options are number A 9, number B 10, number C 5 and number D 7. What is the number of the major classes of enzymes? 9, 10, 5 or 7? Again, I will wait for a minute and then I will check the chat box. Meanwhile, let me know what do you think. What is the correct answer for problem number 3? What is the number of the major classes of this of the enzymes? 9, 10, 5 or 7? What is the number of the major classes of the enzymes? Number A 9, number B 10, number C 5 or number D 7? What is the number of major classes of enzymes? Let me check the chat box. Let me check if there is any answer for this problem. Okay, D is number 7, number 7, number 7, D. Okay, yeah, good evening. So I think I have got all the answers which says that 7 is the answer, correct answer, means number D is the correct option. Now let me check if number D is the correct answer or not. So the correct answer, the number of major classes of enzymes, very good, it is 7. Okay. So naming and classifications of the enzyme is another thing which is very important. So the name of an enzyme reflects the type of the reaction it catalyzes and has the suffix "ase" to indicate that it is an enzyme. For example, an oxidase enzyme catalyzes an oxidation reaction. It is important to appreciate that enzymes can catalyze the forward and back reactions of an equilibrium reaction. This means an oxidase enzyme can catalyze reductions also, as well as the oxidations. This reaction catalysis depends on the nature of the substrate, that whether it is reduced or in the oxidized form. Enzymes classified according to the general class of the reaction they catalyze are coded as the EC number. So EC1 is the oxidoreductases, which catalyzes the oxidations and reductions. EC2 is the transferases, which catalyzes the group transfer reactions. EC3, which are the hydrolases, catalyzes the hydrolysis reaction. EC4 is the lyases, addition or removal of the group from the double bond. EC5 is the isomerases, isomerizations or the intramolecular group transfers, and EC6 is the ligases, joining two substrate at the expense of the ATP hydrolysis. So this was up to 2018. Now in 2000, after 2018, we have another class which are the translocases, a relatively new class added in 2018 that catalyzes the movement of the ions or molecules across the cell membranes or their separation within the membranes. Okay. So enzymes are primarily classified by the International Union of the Biochemistry and Molecular Biology, which is IUBMB, based on the specific chemical reaction they catalyze. This standardized system assigns every enzyme a 4-digit enzyme commission EC number, which is the first digit represents the one of the 7 major classes. One is the oxidoreductases, catalyzes the redox reaction, for example, alcohol dehydrogenase; transferases, which catalyze group transfer like hexokinase; hydrolases catalyze the cleavage of the chemical bond or what by addition of water, which is hydrolysis like lipase, amylase or protease; lyases catalyze the breaking of the very chemical bond like C-C bond, CO-CN bond by the means of hydrolysis or oxidation, or often forming double bond or new ring like aldolase. Isomerase catalyze the structural geometric changes within the single molecule, converting it to the isomeric form like phosphoglucomutase. Then we have ligase catalyze the joining to large molecule to form a new chemical bond, typically coupled with the hydrolysis of ATP like DNA ligase; and translocases, which catalyzes movement of the ions or molecules or their separation within the membrane. So these are the 7 major classes of enzyme. You have to remember what is the EC1, which is the EC2, which is the EC3, the names, the numbers. Also, what does these enzymes do and their examples. So this is very important. This remembering this chart is very important. Okay. So yes, very good. Number D, the 7 was the correct answer. Okay. Now let us move to the next problem. Problem number 4. Which of the following statement is not true for Km and Vmax? Your options are: number A, Vmax is the maximum rate at which a particular enzyme catalyzed reaction can proceed. Number B, a small value of Km tells us that the enzyme binds strongly to its substrate. Number C, Km is the concentration of the substrate at which the rate of the reaction reaches to the Vmax. And number D, a large value of Km tells us that the enzyme shows little specificity for a given substrate. You have to identify the statement which is not true for Km and Vmax. First, you try what is the correct answer. Let me know which of the following you think is the correct answer. Then I will check the chat box. And after that, we will discuss what is Km, what is Vmax, and about these things little. I think you have already learned about this Km and Vmax. So we will now discussing this kinetics part in detail. Okay. Again, let me wait for a minute. Then I will check the chat box. Which of the following statements is not true for Km and Vmax? Number A, Vmax is the maximum rate at which a particular enzyme catalyzed reaction can proceed. Number B, a small value of Km tells us that the enzyme binds strongly to its substrate. Number C, the Km is the concentration of the substrate at which the rate of the reaction reaches Vmax. And number D, a large value of Km tells us that an enzyme shows little specificity for a given substrate. Which of the following statement is not true? Let me check the chat box now. Okay, it's fine. If you don't have any, if you don't know the answer. Okay, so number C, number C, number C, number C, number C, number C, number C. I have got max majorly. I have got number C. Also, I have got one number A and on one number B. Okay, if it is fine perfectly. If you don't know the correct answer, we will be discussing it now. So I have got most of the answers as number C, one number A and one number B. Let us see what is the correct answers of the statement which is not true regarding Km and Vmax will be. Yes, it is number C, which says that Km is the concentration of the substrate at which the rate of the reaction reaches Vmax. Now, we are going to learn something which is known as the Michaelis-Menten curve, and it is a very, very important in case of the enzyme catalysis. Okay, so this is a particular curve for an enzyme catalyzed reaction. On the y-axis, we have the velocity of the reaction, and the x-axis, we have the substrate concentration. Okay, so as you can see, initially, the graph looks like this. Okay, so this is the first order kinetics. Okay, so what happens here? Here lots of enzymes are present. Okay, and as you can see, the substrate concentration is lowest at here and highest in this position. So here the number of available enzymes are constant. Okay, so now when the substrate concentration is low, lots of enzymes are available. So here it follows the first order kinetics. Okay, first order kinetics is followed here. Then at the middle there is a competition between the enzyme and molecule and substrate. Since the substrate, now the number of substrates is also increasing, so here we can see a mixed order reaction. Now when the substrate concentration is very high, means there are lots of substrate and all the, but the enzymes, number of enzymes are constant, so all the enzymes are almost occupied and we have got a saturation, which is the zero order kinetics. This saturation leads to the Vmax, which is the highest rate of the reaction. Okay, and what highest velocity or the maximum rate of the reaction. And when the substrate concentration, so this is this is the Vmax, this is the Vmax by 2, which is the half maximum with the half maximum rate. At this half maximum rate or Vmax by 2, the substrate concentration is known as the Kmax. Okay, so let me just draw it once. So as you can see, this is the Vmax. Okay, so this is the highest constant or highest velocity of the reaction. So this is the Vmax by 2, which is the half of the Vmax. Okay, then if what we have to do is we have to still after getting the Vmax, we have to identify what is the Vmax by 2. It will be on here in the y-axis. We have to draw a perpendicular on the curve, and then we have to draw a perpendicular to. So this curve is not perfect. This curve, you can see, this is more perfect curve. So you can see this. So after you have to draw a perpendicular on the x-axis, you will get a substrate concentration. This substrate concentration, which has the velocity of half maximum, it is known as the Michaelis. This is known as the Km or Michaelis-Menten constant. Okay, so Km is the concentration of the substrate at which the rate of the reaction reaches Vmax by 2, not the Vmax. That is why statement number C is false. Okay. So this is more, I mean, this graph is more better. This graph is better rather than this graph. Okay, as you can see, this is the half of the Vmax. This is the Km, the substrate concentration at half of the Vmax. This is the Vmax. Okay, and this equation is known as the Michaelis-Menten equation. So what is the Michaelis-Menten equation? Michaelis-Menten equation is V0, which is the initial velocity in mole per time. Okay, equal to Vmax into substrate concentration. Vmax is the maximum velocity into the concentration of the substrate divided by Km plus substrate concentration. Okay, where the Km is the substrate concentration as half maxima and Vmax is the maximum velocity. Now, at the first, I have said that if there is I have mentioned in initially when the lots there is a substrate concentration is low, there is a first order kinetics. If this can be detected from this particular slide also. Okay, this particular equation also. So if V0, this is the equation. So if substrate concentration is very low, okay, let's say substrate concentration, we have the substrate concentration. If this substrate concentration is very low, which is the initial stages of the reaction, the substrate concentration is very low rather than the Km value, then what will happen? In the denominator, we can omit the substrate concentration. Okay, we can neglect the substrate concentration if it is very low. So what the equation, what will be the equation? The equation will be V0 by Vmax by Km into the substrate concentration. So this, as you can see, this particular rate of the reaction, it depends on the only the substrate concentration. So this is the first order kinetics. So when the substrate concentration is very low, it follows the first order kinetics. Okay, but when the substrate concentration is very high at the saturation, if the substrate concentration is very high rather than the Km, so in the denominator we can now neglect the Km. So what we will get, we will get V0 equal to V max into the substrate concentration, okay, divided by the substrate concentration. So the substrate concentration, this get cancelled out and we get V0 equal to V max, which is this one. Okay, so now it follows the zero order kinetics. So that is the thing which I have mentioned here. It is also mentioned here that it is follows the first order and second and the zero order kinetics, how it follows. Okay, so this is the basic of the Michaelis-Menten equation. You have to remember the equation and also the graphs, because both the things are very important. Okay, so I hope it is clear. If you have any doubt or any query, let me know in the chat box. I will again repeat it. Okay. Let me go to the next problem. Meanwhile, if you have any doubt, you can let me know. I will discuss it later. Okay. Now, problem number 5 is a mathematical question which says that what is the ratio of Vmax by V0 when the substrate concentration equal to 8 km? Your options are number A 9 by 8, number B 10 by 9, number C 9 by 10 and number D 1 by 8. What is the ratio of Vmax by V0 when substrate concentration equal to 8 km? Number A 9 by 8, number B 10 by 9, number C 9 by 10 and number D 1 by 8. What is the ratio of Vmax by V0 when substrate concentration equal to 8 km? You have to use that particular Michaelis-Menten equation which I have shown just now in the previous slide. If you put the values accordingly, you will get your answer. This is very easy question, but I will give you 2 minutes, then I will check the chat box. Okay. What is the ratio of Vmax by V0 when S equal to 8 km? Just write the Michaelis-Menten equation which I have shown in the last slide. Put the value of S into the equation and you will get the answer. It is very easy. Okay. In today's class, we will be solving few mathematical questions. This is a simple question, okay? But a few questions, they are rather little difficult. So if you do not understand that particular question right now, it is fine. Once I share the slide to you after today's class, you can go through that particular questions or particular mathematical problems by yourself, okay? And then in the next class, in the upcoming class, you can let me know if you have any doubt or query regarding the mathematical questions, okay? Because in the short span of time it is not possible for me to show each and every question in detail to solve the questions. I have solved the questions in detail, but you have to go through them once before understand. Otherwise, I think there might be some problem in understanding. So if you have any problem in the mathematical questions in today's class, let me know via email or let me know in the next class. I will surely solve them again for you. Okay. So what is the ratio of Vmax by V0? Let me know. Let me wait for another minute, then I will check the chat box. What is the ratio of Vmax by V0 when S equal to 8 km? Let me check the chat box. Let me see if there is any answer. Okay, very good. I have got lots of answers. Number A, number A, number A. So everybody thinks it is number A is the correct answer. Let me check if number A is the correct answer or not. So the correct answer will be very good. It is number A. This is very simple. I have already told you. So this is the Michaelis-Menten equation, right? V0 is equal to Vmax into substrate concentration divided by Km plus S, where V0 is the initial velocity in mole per time, S is the substrate concentration in molar, Vmax equal to maximum velocity, and Km is the substrate concentration at half Vmax. Okay. So, according to your question, substrate concentration equal to 8 km, right? So, if we take Vmax into the left hand side, we will get V0 by Vmax equal to substrate concentration divided by Km plus S, right? So, now you have to substitute the substrate concentration or S by 8 km. So, if we do that, we will get 8 km divided by Km plus 8 km, which is 8 km divided by 9 km. Km cancels out, we have 8 by 9. Now you have been asked to find out the ratio of the Vmax by V0, which will be 9 by 8. Okay, so this is the answer. This is very easy, very simple. Only you have to put the values into the equation and you will get the answer. Okay, so I have got all the correct answers, so very good. Now let me go to the next problem. Problem number 6 is, which coenzyme is required by the enzyme lactate dehydrogenase? Your options are number A NADP, number B NAD+, number C NADH, and number D FAD+. Which coenzymes is required by the enzyme lactate dehydrogenase? NADP, NAD+, NADH, or FAD+? Again, I will wait for a minute or two, then I will check the chat box. Let me know which coenzyme is required by the enzyme lactate dehydrogenase. After checking the chat box, I will discuss it in detail, okay, regarding what is the enzyme lactate dehydrogenase, what is the coenzyme, all the things I will be discussing, but first you have to try to answer, okay? Which coenzyme is required by the enzyme lactate dehydrogenase? Let me check the chat box. Let me see if there is any answer for problem number 6. Okay, I have got many answers. Okay, one number A, NAD plus, number B, number B, number B. Okay, so let me check. Number B is the maximum, which the answer which I have got. Let's see if it is the correct answer or not. So the coenzyme which is required by the enzyme lactate dehydrogenase, it will be, yes, very good. It is number B, NAD plus. First, let us see what does this lactate dehydrogenase do. So the lactate dehydrogenase, it converts the lactic acid to pyruvic acid. So what is the reaction, as you can see? So basically it is a reduction reaction, right? So dehydrogenase means it has eliminated two hydrogens from our system. So as you can see, lactic acid, it has one hydroxyl group and hydrogen in this carbon position. So the enzyme, has a nucleophilic center which abstracts this proton from the hydroxyl proton. There is double bond is from, and this hydrogen goes there. Okay, so this is the reaction where NAD plus is the coenzyme. NAD plus is reduced to the NADH. Okay, so this is the structure of the NADH. So here NAD plus is acting as a coenzyme for the lactate dehydrogenase. Dehydrogenase, again, this name suggests that it catalyzes this dehydrogenation kind of reaction. Okay. So now let us understand what is the coenzyme, what is the cofactor and everything. Okay. So many coenzymes require additional non Many enzymes require additional non-protein substances called cofactor for the reaction to take place. Deficiency of cofactors can arise from the poor diet, resulting in the loss of the enzyme activity and subsequent disease like scurvy. Cofactors are either metal ions like zinc or small organic molecules called coenzymes. Now, for example, NAD plus pyroxyl phosphate. Most coenzymes are bound by ionic bond or other non-covalent bonding interactions, but some are bound covalently and are called the prosthetic groups. Coenzymes are derived from the water-soluble vitamins and act as body's chemical reagents. For example, lactate dehydrogenase requires the coenzyme nicotinamide adenine dinucleotide or NAD+ in order to catalyze the dehydrogenation of the lactic acid to pyruvic acid. NAD+ is bound to the active site along with the lactic acid and acts as an oxidizing agent. During the reaction, it is converted to its reduced form NADH. Conversely, the NADH can bind to the enzyme and act as a reducing agent when the enzyme catalyzes the reverse reaction. As you can see, this is the equilibrium reaction. So, for the reverse reaction, NADH can act as a coenzyme. NADP plus and NADPH are the phosphorylated analogues of NAD plus and NADH respectively and carry out the redox reaction by the same mechanism. NADPH used in most exclusively for the reductive biosynthesis, whereas NADH is used primarily in the generation of ATP. So what is the differences between a coenzyme and cofactor? So coenzyme is a small organic non-protein molecule that carry chemical group between the enzymes. Cofactor is a non-protein chemical compound that tightly or loosely bounds with an enzyme or other protein molecules. Coenzymes is a type of cofactor. Coenzyme and prosthetic group are two types of cofactors. Coenzymes are basically molecules and cofactors are the chemical compound. Coenzymes are organic molecule, cofactors are inorganic compounds. Okay, loosely bound to the coenzyme closely bound to the enzymes. Cofactors, some are covalently bound to an enzyme. Coenzyme assist the biological transformation and serve as carriers to the enzyme, whereas the cofactor aid the function of the relative enzyme, increase the rate of the reaction that is catalyzed by the enzyme. Coenzyme can be removed from the enzyme easily. For example, at the vitamin biotin coenzyme A. The cofactors can only be removed by denaturing the enzyme. For example, include the metal ions like zinc plus 2, K plus or magnesium plus 2. Okay. So these are the differences between the coenzymes and cofactor. And this is the answer was correct. So you have to remember what is the role of the lactate dehydrogenase this reaction. Also this table which contains the differences between the coenzyme and cofactor. Okay. So now let us go to the next problem. Problem number 7, again it is a mathematical question. It says an enzyme has a Km value of 7 millimolar and the rate of the reaction is 150 millimole per minute when the substrate concentration is 0.5 millimolar. When the enzyme is saturated with the X substrate, what is the maximum rate of the reaction? Means you have to find out the Vmax. The Km value is given, the rate of the reaction V0 is given 150 millimole per minute when the substrate concentration S is 0.5 millimolar. Now you have to find what is the Vmax. Your options are number A 2500 millimole per minute, number B 1100 250 25 millimole per minute, number C 1250 millimole per minute and number D 2250 millimole per minute. Write what is the Michaelis-Menten equation and then put all the values and you will get the answer. I will give you 2 minutes, then after that I will check the chat box. Okay, try it by yourself, use a pen or paper, try it by yourself and let me know A, B, C or D which of the following will be the correct answer. Try to solve the question. Okay, I will wait for another minute, then I will check the chat box. The Km value is given, rate of the reaction is given, which is the V0, and you have the substrate concentration. What you have to find out is the Vmax value. Just put the values in the Michaelis-Menten equation and you will get the answer, okay? Let me check the chat box. I hope I will find some answers there. Okay, very good. I have got a lots of answers. So most of the answers are saying that number D is the correct answer. Let me check if number D is the correct answer or not. Okay, and let us see how to solve this kind of question. So the correct answer will be very good. It is number D. 2250 millimole per minute is the maximum rate that the reaction can achieve. Again, I have said only you have to follow this Michaelis-Menten equation, which is V0 equal to Vmax into S divided by Km plus S. Okay, all the values are given in the question, right? The velocity mole per time is given here like 150. So V0 value is 150 mole per minute. A substrate concentration is 0.5 millimolar and Km is also given as 7 millimolar. So according to the problem, Km is 7 millimolar, a substrate concentration S is 0.5 millimolar when V0 equal to 150 millimole per minute. Now only you have to substitute the values into the this equation. So V0 we know equal to Vmax into S divided by Km plus S. If we take Vmax to this side, and so we will be getting Vmax equal to V0 into Km plus S, okay, divided by substrate concentration. So V0 is 150 millimole per minute into 7 plus 0.5 millimolar divided by 0.5 millimolar. So what we will get, millimolar cancel out, and if you calculate the values, you will get the value 2250 millimole per minute. Hence, the maximum rate achieved by the enzyme is 2250 millimole per minute. Okay. This is very easy. If you have any doubt or any problem in any mathematical question, I will suggest try it by yourself. Once I share the slides, please try it by yourself once at your home, okay? And after that, you can let me know via email if you have particular doubts in particular question like problem number 7. If you do not understand after trying it by yourself, once you provide you the PPT, then you can let me know via email. I will solve the particular question again in the upcoming class. Okay, and in the upcoming class also itself you can let me know that you have any problem in any mathematical question. Okay, so I have got answers for this question. So I hope it is clear, right, how we have solved it. If you have any doubt or query, you can let me know now. So you have understood this problem. I have got the correct answer. So I hope that okay, very good. Thank you for letting me know that it is clear. So this is very easy. I think if you have any, again I am saying try it by yourself. Once I provide the PPT, then it will be more clear. Okay. Now let me go to the problem number 8. This is also a mathematical question. Okay, it says consider the following values of Michaelis-Menten enzyme kinetics and calculate the dissociation constant for an enzyme substrate complex and catalytic efficiency for an enzyme. The values are given K1 equal to 4 into 10 to the power 8 millimole per minute. Okay, inverse the mole sorry, it's molar per minute inverse. K minus 1 equal to 1 into 10 to the power 3 minute inverse and K2 equal to 6 into 10 to the power 3 minute inverse. Okay. Your answers are number is 0.5 micromolar, 8 is the for dissociation constant, and for catalytic efficiency it 8.57 into 10 to the power 8 molar per minute inverse. Number B is this one 2.5 micromolar and 8.57 10 to the power 8 mole per minute inverse. Number C is 2.5 micromolar and 3.42 into 10 to the power 8 molar per minute inverse, and number D 5 micromolar and 0.857 into 10 to the power 8 molar per minute inverse. So what will be the correct answer? This is a little different question. Okay, try to solve it. I will show you how to do it, so you don't have to worry. I will give you 2 minutes, okay? Let's see if you can do it within 2 minutes. Otherwise, then I will show you how to do it, okay? Just let me tell you what is the K1, K minus 1 and K2. So when we have a particular, let's say we have a particular substrate, okay, and we have an enzyme, right? So the first thing which in the first step of the reaction, it is a equilibrium. Okay, it is a two-way reaction. The forward reaction rate constant is K1 and the backward reaction K constant is K minus 1. Okay, it forms the enzyme and substrate complex. This enzyme substrate complex is dissociated to the product, and the rate constant of this particular step is the K2. Okay, so this K1, K minus 1 and K2 are these rate constant for this particular thing. So that is what I have to tell you. I will wait for some time, then I will show you the answer. Okay, try, let's see if you can do it by yourself. Again, I am saying some answers they might seem to be little complicated, but once you do it by yourself, by yourself, once I share the PPT, you will see these are the very simple questions, simple and but just you have to know the particular formulas and you have to put the values and the units correctly. So let me check if there is any answer for this problem number 8. Okay, I got two answers and both are saying that number B is the correct answer. Okay, let me check if number B is the correct answer or not, and then I will show you how to do this particular, how to solve this question. So the correct answer is, it is number C. Okay, 2.5 micromolar and 3.42 into 10 to the power 8 millimole per minute inverse. So now this formulas which you have to remember. Okay, so this first is this thing. First is this equation that I have not mentioned it here. So the dissociation constant for the enzyme substrate complex is given by your K minus 1 divided by K1. Okay, and the catalytic efficiency is given by K2 by Km. So these are two formulas that you have to remember. First is that dissociation constant for a enzyme substrate complex is given by K minus 1 divided by K1. Okay, so the values are given. The K minus 1 is 1 into 10 to the power 3 minute inverse and K1 equal to 4 into 10 to the power 8 molar per minute inverse. So if you do it, if you divide these two, so you will get that the answer is 0.25 into 10 to the power 5 molar or 2.5 micromolar. Okay. And then catalytic efficiency is given by K2 by Km. Now Km is the Michaelis-Menten constant. Okay, so this K will be capital. Okay. Yeah. So this Km is the Michaelis-Menten constant. So the Michaelis-Menten constant again is given by this formula. Km equal to K minus 1 plus K2 by K1. Okay. So if you drop all the values here, you will be getting the Km value like 1.75 into 10 to the power minus 5 molar. And then catalytic efficiency is given by K2 divided by Km, which is K2 is this thing and Km is this. And if you divide this two, you will get the answer like this. Okay. It may seem a little complicated, but once you do it by yourself, once you put the values by yourself, you will find that this is not very complicated. This is very easy actually. Okay. Just you have to put the values correctly to the formula. Then for that you have to know what are the particular formulas, right? So these are the things which you have to remember first. What is the km? How to that is the thing which you need to identify. Okay, what is km? And then you have to identify. Let me just correct few things. Yeah. So these are the things which you need to remember. What is the things that the dissociation constant is given by K minus 1 by K1, catalytic efficiency is given by K2 by Km, and Km is given by K minus 1 plus K2 by K1. All the values are given. Only you have to put the values and you have to take care of the units also. Okay, be careful. Beware of the units also. Sometimes because of units, some answer may be correct or incorrect. Okay. I know this might seem a little complicated, but I promise if you try it by yourself, you will find this very easy. Okay. And still, if the problem persists, let me know in the next class or via email. I will repeat this particular problem. Okay. Let us go to the next problem then. So the problem number 9 says which type of catalysis is seen in presence of histidine? Your options are number A electrophilic, number B nucleophilic, number C acid base, number D all of the above. Which type of catalysis is seen in presence of histidine? Electrophilic catalysis, nucleophilic catalysis, acid base catalysis or all of the above? Again I will wait for a minute, then I will check the chat box. Let me know which type of catalysis is seen in presence of histidine amino acid. Which type of catalysis is seen in presence of histidine? Electrophilic, nucleophilic, acid base or all of the above? I have shown you the structure of histidine in our previous class. I think when I was discussing about the table containing all the amino acids and the classification, histidine falls under a particular class of amino acid. If you can remember that, I think you can easily answer this question. What type of catalysis is seen in presence of histidine? Let me check the chat box. Okay, I got number D, number D, number C, number D, number B also I have got. So I have got basically three different options as answer. Number B, number C and number D also. Okay, okay, let me check which of the following will be the correct answer for problem number 9. So the type of catalysis which is seen in histidine will be, it is number C, acid-base catalysis. Now, why I have told you that to remember what class of amino acid is histidine. So, basically histidine we all know is a basic amino acid. Okay, and the histidine, why it is a basic amino acid? Because of the presence of this imidazole ring. Okay, so acid-base catalysis is often provided by the amino acid histidine, which contains an imidazole ring as a part of its side chain. The imidazole ring, this is the imidazole ring, a 5-member heterocyclic ring which contains two nitrogen in 1 and 3 position. It is known as the imidazole. These two nitrogen have two lone pair, which two lone pair can have this kind of resonance stabilization of the charge. Okay, and this histidine, this works as a weak base. The imidazole ring reacts as a weak base, which means it exists in equilibrium between its protonated and free base form, allowing it to accept or donate proton during a reaction mechanism. This is important as there are often few water molecules present in the active site to carry out this role. Histidine is not only amino acid that can provide acid-base catalysis. For example, glutamic acid residue acts as a proton source for the reaction reaction mechanism of the enzyme HMG-CoA reductase, while aspartic acid and aspartate residues act as a proton donors and proton acceptors respectively in other enzyme catalysis reaction. Tyrosine acts as a proton source for the mechanism by which the enzyme 17-beta hydroxysteroid type 1 catalyzes the conversion of estrone or estradiol. So these are the examples of the acid-base catalysis. Okay, now let me highlight the things because uh, these are sometimes question can be given with the other amino acids also. So for glutamic acid, remember it acts as a proton source in the mechanism of action for the enzyme HMG-CoA reductase. This about regarding this enzyme HMG-CoA reductase we will be discussing in detail uh, in our future slides because we have this particular topic in your syllabus. While aspartic acid and aspartate residues act as a proton donor and acceptor in other enzyme catalyzed reaction, tyrosine acts as a proton source when the mechanism by the for the enzyme 17-beta hydroxysteroid type 1, which catalyzes the conversion for the estrone to estradiol. Okay, so these are the examples of the acid base catalysis. Okay, so so the number C was the correct answer, and I think in the chat box I have seen there was a number C. So very good, whoever has answered that question. Now let me go to the next problem. So problem number 10 says the enzyme cyclase is activated by dash. Fill in the blanks. Your options are number A ATP, number B water, number C NO2 and number D NO. Well, in the enzyme cyclase is activated by what? ATP, H2O, NO2 or NO. Again, let me wait for a minute or two, then I will check the chat box. Let me know A, B, C or D, which of the following you think is the correct answer. See, if your answer is wrong, it is fine, but please answer, otherwise I won't understand you are following the class or not, okay? Even if your answer is wrong, it is perfectly fine, but at least try to answer. If you have a slight idea regarding the particular question, please try to answer. The enzyme cyclase is activated by what? ATP, H2O, NO2 or NO? The enzyme cyclase is activated by what? ATP, H2O, NO2 or NO? Let me check if there is any answer for problem number 10. Okay, I think I have got answers. Number A, number A, number D, number A, number A, number A. The screen share is visible. The screen is visible to all, or it is not. Can somebody let me know if the screen share is still on, or is there any problem? The slides are visible, right? Okay. So Sujata, I think you have an issue with your network. You can rejoin, then you can see the slide. Yes, histidine can act as a nucleophile, but the basicity of this particular histidine is greater than the nucleophilicity. Okay, the majorly this histidine catalyzes the acid base catalysis, not for the nucleophilic catalysis. Okay, so yeah, the slides are now visible, I think. So number A and number D. So I have got maximum answer as number A and few answer one as number D. Let me check which of the following is the correct answer. So the enzyme cyclase is activated by, very good, by NO. The enzyme cyclase is activated by NO. Okay. So this is known as the external control of the enzymes. Okay. So first let us see what are the roles of nitric oxide. So nitric oxide or NO, it mediates the macrophage cyclotoxicity against the microbe. It inhibits the platelet aggregation. It Then it protects brain tissues via the hemodynamic regulation, induces vasodilation, and increases the blood flow. It is a mediator for the migraine and other vascular headaches, and protects the muscular lining of the gastrointestinal system. Okay, so these are the roles of nitric acid in our body. So this is known as the external control of the enzyme. So first, let us see how the nitric oxide is synthesized. So basically, we have so the external control of enzyme is usually initiated by the external chemical messengers which do not enter the cell. However, there is an exception to it. It has been discovered that the cells can generate the gas nitric oxide by the reaction sequence shown in 1, which is catalyzed by the enzyme nitric oxide synthase. So this we have the arginine. Arginine is another basic amino acid which has its guanine side chain. This arginine can form this citrulline, okay, by this enzyme with the help of the anosynthase enzyme, okay, and NADPH converts to NADP plus in this role of the this phases or steps of the reaction. Okay, so when arginine is converted to the citrulline by this enzyme anosynthase, nitric oxide or NO is formed. Okay, this nitric oxide is a gas. It can diffuse easily through the cell membranes into the target cell. There it activates enzymes called cyclase to generate the cyclic GMP to GDP. The cyclic GMP acts as a secondary messenger to influence other reaction within the cell. By this process, nitric oxide has an influence on the diverse range of physiological process, including blood pressure, neurotransmission, and immunological defense mechanism. Okay, so all these things which can be controlled by nitric oxide, and how? Because nitric oxide enzyme that activates the enzyme cyclase, which generate the cyclic GMP, which has a very crucial role in the cell. Okay, so this is the guanosine triphosphate or GTP. They're similar as ATP, but only this base is not adenine, it is guanine. Okay, and this GTP is converted to cyclic GMP or cyclic guanosine monophosphate. Okay, by the help of the cyclase enzyme, which is activated by the nitric oxide. So the enzyme cyclase is activated by the nitric oxide. Remembering the structures is not very much important. If you can remember, it is very good. If it is not, it is fine, but try to remember which reaction it is catalyzing. Okay, like the cyclase is catalyzing the conversion of GTP to GMP, and NO is acting as the activator for the cyclase. So these things you have to remember. Also remember the synthesis of the nitric oxide, how it is synthesized into the cell. Okay, so there was a cancer number D, NO, which is very good. Okay. Now let me go to the next problem. Problem number 11 says, what is the cyclic triad of amino acids in the active site of the enzyme chymotrypsin? Your options are number A, serine, arginine and glutamate. Number B, threonine, histidine, methionine. Number C, serine, histidine and aspartate. And number D, threonine, arginine and glutamate. What is the catalytic triad of amino acid? Catalytic triad of amino acid means 3 amino acids which are important for the catalytic activity of the chymotrypsin. Okay, in the active site of the enzyme of chymotrypsin. Your number A, serine, arginine, glutamate. Number B, threonine, histidine and methionine. Number C, serine, histidine, aspartate. And number D, threonine, arginine and glutamate. Again, I will wait for a minute, then I will check the chat box. Let me know A, B, C or D, which of the following is the correct answer. What is the correct set of catalytic triad for the active site of chymotrypsin? What is the catalytic triad of amino acid present in the active site of chymotrypsin? Number A, serine, arginine, glutamate. Number B, threonine, histidine, methionine. Number C, serine, histidine, aspartate. And number D, threonine, arginine and glutamate. Let me check the chat box. Let's see if there is any answer for problem number 11. Okay, I have got many answers. Number C, number C, number B, number C, number C, number C. Okay. Number C is the most given answer, and I got one number B. Let me check which of the following is the correct answer. Majority is toward number C, so let's see that if it is correct answer. So the correct answer will be very good. It is number C. Serine, histidine, and aspartate is the catalytic triad of amino acid present in the active site of the chymotrypsin. So this is the active site of chymotrypsin. Okay, so the amino acids serine, cysteine are present in the active site of some enzyme. These amino acids have nucleophilic OH and SH respectively. So now if last in you have said that histidine can act as a nucleophile. It is true that histidine has some kind of nucleophilicity, but majorly the histidine acts as a acid base catalysis, and it actually basically it abstracts the proton, and at the time it donates the proton. Okay, but the amino acids like serine and cysteine, which has this OH and SH, they acts as a nucleophilic residues. Okay, this OH and SH acts as a nucleophilic residue. So how this reaction happens? So basically, chymotrypsin, what does it do? It cleaves the peptide bond. Okay. So how does it do? Here you can see there is a serine. Serine has a nucleophilic OH. This OH attacks to the carbonyl carbon of this our peptide linkage. Okay, and there this here in presence of histidine and aspartate acid, basically this aspartate residue activates the histidine, and histidine abstract this proton from this OH to make this hydroxyl group a better nucleophile, right? Then what does it do? Again, when there is a this double bond is formed, it cleaves the bond, and it acts as a proton donor. So first it acts as a proton acceptor. Histidine here it acts as a proton acceptor, and here it acts as a proton donor. Okay, so that's how this catalyzes the acid base catalysis, and serine is there for the for making the nucleophilic bond. Now water molecule comes and it hydrolyzes basically this residue, and we get our cleaved form, cleaved OH. Here's the serine and the histidine as it is. Okay, so the amino acid serine and cysteine are present in the active site of some enzyme. These enzymes, amino acids, have nucleophilic residues OH and SH respectively, which are able to participate in the reaction mechanism. They do this by reacting with the substrate to form the intermediates that would not be formed in the uncatalyzed reaction. These intermediates offer an alternative reaction pathway that may avoid a high energy transition state, hence increase the rate of the reaction. Normally, an alcoholic OH group such as one in the serine is not a good nucleophile. However, there is usually a histidine residue close by to catalyze the reaction. For example, mechanism by which chymotrypsin hydrolyzes the peptide bond involves a catalytic triad of amino acid, histidine, serine, and aspartic acid. Serine and histidine participate in the mechanism as a nucleophile and acid-base catalyst respectively. The aspartate group interacts with the histidine ring and serves to activate and orient it correctly to the mechanism. The presence of the nucleophilic serine residue means the water is not required in the initial stages of the mechanism. This is important firstly because water is a poor nucleophile and may also find it difficult to penetrate to occupied active site. Secondly, a water molecule would have to drift into the active site and search out for the carboxyl because it could attack it. This would be something similar to the game of let us say so the enzyme however can provide the serine OH group possibly position exactly the right position to react with the substrate. Therefore, nucleophile has no need to search for its substrate, the substrate being delivered to it. Water is eventually required to hydrolyze the acyl group attached to the serine residue, this one. Okay, and this however this is a much easier step than the hydrolysis of a peptide linkage, as esters are more reactive than amide. Furthermore, the hydrolysis of the peptide linkage means one half the peptide can drift away from the active site, leaving the room for water molecule to enter. A similar enzymatic mechanism is involved in the action of the reaction acetylcholine transferase, pancreatic lipase, and the viral protease enzyme carried by hepatitis C virus. Okay. So this is the mechanism of chymotrypsin. All this is the details are given. You can read it by yourself and for better understanding. But this is a very simple mechanism. Okay. But this mechanism is very important. So try to practice it by yourself and to check if you can do it do it easily. Okay. Now let me go to the next problem. Problem number 12 says which kind of inhibitor glucose 1-phosphate is for the enzyme phosphorylase A. Your options are number A reversible, number B allosteric, number C suicide, and number D irreversible. What kind of inhibitor glucose 1-phosphate is for the enzyme phosphorylase A? Reversible, allosteric, suicide, or irreversible? Regarding the enzyme inhibitors, the kinetics of enzyme inhibition, all these things in detail we will be learning in our upcoming class, means in our week 3. Okay. But I thought to give you a little one or two question regarding this in today's class also. So what kind of inhibitor glucose 1 phosphate is for enzyme phosphorylase A? Enzyme inhibition actually are various types and various classifications are there regarding the enzyme inhibition. All these things we will be learning in our upcoming week 3 in more detail. Okay. Let me wait for a minute, then I will check the chat box. What kind of inhibitor glucose 1 phosphate is for enzyme phosphorylase A? Let me check the chat box. Okay, there are few answers. Reversible, number B, number C, number A. Okay, I think I have got four different options as answer. Number A, B, C, and D also. Okay, it's fine if you do not answer it correctly, because we will be discussing regarding enzyme inhibition in our upcoming class. Okay, so the correct answer will be, it is number B, allosteric. Okay. So there are different kind of enzyme inhibitions are possible. Some enzyme inhibitors bind itself to the active site so that the substrate cannot bind. Okay, some enzymes they bind to a different site rather than the active site and changes the structure of the active site. These kind of inhibitors are known as the allosteric inhibitor. Again, I am saying we will be discussing regarding this enzyme inhibition in detail in our upcoming class. Just I thought to give you a little brief about it. So allosteric inhibitors are the molecules which bind to a different site of the enzyme, not in the active site, and conformationally the active site changes once this allosteric inhibitor binds. The active site becomes shut and the substrate cannot bind. Okay, so this is known as the allosteric inhibition of enzyme. This same thing happens when the same thing happens with the phosphorylase A, the catalytic activity of phosphorylase A by glucose phosphate and AMP. So virtually all enzymes are controlled by agents which can either enhance or inhibit their catalytic activity. Like NO, the nitric, in our last to last slide, in the nitric oxide, it can enhance the activity of that particular enzyme cyclase. Here, in case there are some reagents or some uh, agents which can inhibit the catalytic activity. For example, enzyme phosphorylase A, this phosphorylase A catalyzes the breakdown of the glycogen. This is the glycogen, a polymer of glucose monomer, to glucose 1 phosphate subunit. This is glucose 1 phosphate subunit. It is stimulated by the adenosine 5 prime monophosphate or AMP and inhibited by the glucose 1 phosphate. Thus, the rising level of the product is acts as a self-regulating brake on the enzyme. But how does this control take place? The answer is the enzyme have a different binding site which is separate from the active site called allosteric site. This is where the agents controlling activity of the enzyme bind. When this occurs, an induced fit takes place, altering the active site, and agents that inhibit the enzyme produce an induced fit that makes the active site unrecognizable for the substrate. Okay, so basically for this particular phosphorylase A enzyme, it is activated by this AMP and inhibited by the product GT, this glucose 1 phosphate. This glucose 1 phosphate binds to allosteric site of the enzyme and modifies the active site such as the substrate now cannot bind. Okay, so this is also known as the allosteric inhibition. Okay, so that is how. So basically, you have to remember that there will be agents that can either induce the enzyme, or they can enhance the enzyme activity or inhibit the enzyme activity. Nitric oxide was one example. AMP is another example which activates this phosphorylase A, and also inhibitors are another thing. Inhibitor, there is a vast classification, and we have to discuss regarding the inhibitors in detail, and that we will be doing in our upcoming class. Okay, let us go to the next problem. Problem number 13 says anchimeric assistance is commonly known as dash. You have to fill in the blanks. Your options are number A, neighboring group elimination, number B, electrophilic substitution, number C, addition elimination, and number D, neighboring group participation. Anchimeric assistance is known as what? Neighboring group elimination, electrophilic substitution, addition elimination, or neighboring group participation. What will be the correct answer for problem number 13? Anchimeric assistance is commonly known as what? I know all of you can answer it correctly, so I will be waiting for only a minute, then I will check the chat box, okay? Your options are number A, neighboring group elimination, number B, electrophilic substitution, number C, addition elimination, and number D, neighboring group participation. What will be the correct answer for problem number 13? Let me know. Let me check the chat box. I know all of you have answered it correctly. Okay. Majority is toward D. I have got one number A. Otherwise, I have got all number D as the answer. Let me check if number D is the correct answer. Since it is given by majority, I think it is the correct answer. Let me check. So, anchimeric assistance is commonly known as, yes, number D, neighboring group participation. So what is the neighboring group participation? So basically anchimeric assistance is a process by which a neighboring functional group can assist an expulsion of a living group by an intermediate covalent bond formation. For example, if you have a sulfur here, it can attack to this position and help to chloride to leave. Okay, and then the upcoming nucleophile can attack this position and we can get the product. So if the sulfur atom was a part of an active site of a nucleophile such as methionine, the CCl bond was a part of a substrate, the arrow were the generated by the enzyme catalysis reaction. This would represent a covalent catalysis or enzyme catalysis reaction where the covalent adduct is represented by the episulfonium intermediate. So let us see a more elaborated fashion. So neighboring groups can accelerate the substitution reaction. So let us compare the rate of the following reaction. Each one is a substitution of the leaving group OTS or chloride by solvent known as the solvolysis. Okay. So this particular compound reacts with water 600 times faster than this. This reacts with the CF3, the triflic acid 3000 times faster than this. Okay. This reacts with acetic acid 10 to the power minus 11 times faster than this one, and this reacts with the acetic acid 670 times faster than this one. Now why this happens? Okay, so basically because of this neighboring group. So this sulfur has a lone pair which can assist the elimination of this chloride. Okay, but here we do not have any this kind of lone pair present. That is why this reacts faster. Here the phenyl group, this has the double bond of the phenyl group, can assist this particular reaction and elimination of the OTS. Here also this double bond can assist the elimination of OTS. Okay, but here there is no double bond which can assist this reaction. Here also the same, if you can draw this compound in a chair formation, you will see the acetate will assist the elimination of this OTS but not this one. So neighboring group evidently increase the rate of the substitution reaction significantly. Now So this reacts faster than this one because of this O. And so the four examples above, it is not a reaction center itself. Functional group happens the carbon next to the reaction center. So as you can see, this is the reaction center carbon, but the reaction happens with the functional group which is present to the carbon next to the reaction center. We call this group as neighboring group, and the mechanism by which they speed up the reaction is known as the neighboring group participation. Okay. So, for example, this is in both cases the ionization of a starter. So, this is the neighboring group participation. This is not okay. In this case, this group is next to the is attached with the reaction center itself. Okay. So, this is the ionization reaction of the lone pair. The ether is the first example, assists the forming by pi bond. Okay, but here the sulfur acts as a neighboring group and forms this three-member ring intermediate and then induces the reaction. So this is the example of a neighboring group participation. This is not okay. So you have to remember this neighboring group participation. For this neighboring group participation, this the neighboring group must be to the carbon next to the reaction center, not on the carbon itself. Okay. In our enzyme catalysis reaction, in many places we see this neighboring group participation. The examples. Okay, and this is known as the anchimeric assistance. Okay. So now let me go to the next problem. Problem number 14, again it is the mathematical question. Okay, consider the following plot. Okay, the plot is not there. So this is the normal Lineweaver bark plot. Okay, of 1 by V0 versus 1 by S, plot of the Lineweaver bark. The result obtained of the graph is the intercept value is 0.2 micromolar minute inverse and the slope is 7 into 10 to the power 5 minute. What is the km of the reaction? Your options are number A 35 into 10 to the power 5 micromolar, number B 0.258 into 10 to the power 5 micromolar, number C is 7 into 10 to the power 6 micromolar, and number D is 0.011 molar. Lineweaver bark plot is nothing but a different representation of the Michaelis-Menten plot, okay, which is also known as the double reciprocal plot. First, you try to solve the question, then I will show you in elaborated matter what is Lineweaver bark plot, how it is the equation is forming, and how this type of question can be solved. First try. Then I will show you the answer, okay? The intercept value is given, the slope value is given, you have to find out the Km value. One hint I can give you that just first write the Michaelis-Menten equation, make it the reciprocal, okay? Then the rearrange this thing like 1 by V0 and 1 by S. If you know what is the Lineweaver bark plot, what is the equation, you can easily solve this. If you don't know, it is fine. I will show you how to do it. First, let me wait for 2 minutes, then I will check the answer, okay? What is the Km value of the reaction? If the intercept value is 0.2 and the slope is 7.5, if 10 to the power 5 in case of Lineweaver bark plot. I will wait for another minute, then I will check the chat box, okay? Okay, let me check if there is any answer for problem number 14. Otherwise, I have to show you in detail. Okay, I have got few answers. number D, number D, number A, number D, number A, number B. So three different options I have got as answer. Number A, B, and number D. Okay, let me check. It's good that you have tried. Very good. Let me now check the answer. So the correct answer will be, it is number A. Okay, 35 into 10 to the power 5. Now let me show you. So basically Lineweaver bark plot is this one. Okay, so this is the Lineweaver bark plot equation. So now this is very easy to find out the equation from the particular Michaelis-Menten equation. So let me show how you have to find out. So basically in the so where should I do it? Okay, let me just have another slide added here. Just let me just minimize this one a little bit. Okay, this will be fine. So let us see how this we can derive this Lineweaver bark plot from the Michaelis-Menten equation. So the Michaelis-Menten equation we know equal to V0 equal to Vmax. Let us write Vmax as Vm into S divided by Km plus S. If you just take the reciprocal of this equation, now what we will have? We will have 1 by V0 equal to Km plus S divided by V max into S, right? So this is what we will got if you just take the reciprocal. Now if we rearrange this thing, then we will have 1 by V0 or 1 by rate equal to Km. Just I will just break it. Okay, so we will get Km by Vmax into concentration of sorry, just let me erase this thing. Km by Vmax into 1 by S. Okay, then with I will get plus SS will be getting cancelled out, so 1 by Vmax. So this is the equation which is written here. This is the same thing and it is derived from the Michaelis-Menten equation itself. Just you have to take the reciprocal of Michaelis-Menten equation and rearrange. And if you compare it with y equal to mx plus c, then the plot of the Lineweaver bark is like this: 1 by rate or 1 by v0 divide and versus 1 by s. Okay, 1 by s, this is the plot. And this, if this y, this is x, then if you compare it with y equal to mx plus c, m is the slope, you will get Km by Vmax is the slope and the okay, and thus intercept is 1 by Vmax. Okay, so this is the Michaelis-Menten equation and that is how you have to derive this thing. So the Lineweaver bark plot associated with this particular, let me just highlight the things. Okay, so the Lineweaver bark plot equation is this one: 1 by V0 equal to Km by Vmax into C. This is the Lineweaver bark plot. Now, so if you plot this thing, we will get the information intercept is 1 by Vmax. Okay, and the slope is Km by Vmax. Now, according to the question, the intercept value is given. Okay, so according to the question, the intercept value is given to you. The intercept value is 0.2, right? Micromolar per minute inverse, and Vmax equal to 5 into micromole per minute. So the slope will be Km by Vmax and Km will be slope into Vmax. Vmax is given, slope is given, so you have to just multiply and you will get the answer, which is the Km. If you can remember this equation, it is fine, but if you sometimes we forget the mathematical equations, so it is the best way. That is how I remember this thing. Only write the remember Michaelis-Menten equation, write the Michaelis-Menten equation and make the reciprocal. Okay, that's how you will get the answer. So this is very easy. Again, I will tell you that please practice it once I share the presentation. If you have any problem, let me know in the upcoming classes. Okay. Now let me go to the next problem. Problem number 15 says, which one of the following statement is false? Your options are: number A, both receptors and enzymes have active site. Number B, only enzymes has active site, not the receptors. Number C, after binding, reaction takes place in the enzyme. Number D, after binding, no reaction takes place in receptors. Which of the following statement is false? Number A, both receptors and enzymes have active site. Number B, only enzymes have active site, not the receptors. Number C, after binding, reaction takes place in the enzymes. Number D, after binding, no reaction takes place in receptors. Let me tell me which of the following statement will be false. I will wait for a minute. I will be little quick from now on because we have around 6 questions left and which we have to finish it by half an hour. Okay. So which of the following statement is false? Read the options and let me know. I will check the chat box within a minute. Number A, both receptors and enzymes have active site. Number B, only enzymes have active site, not the receptors. Number C, after binding, reaction takes place in enzymes. And number D, after binding, no reaction takes place in the receptors. Which of the following statement is false? Let me check the chat box. Okay, number B, number B, number B. So the maximum answers which I have got, all are saying that this number B is the first statement is false. Okay, let me check if number B is the correct answer or not. So the correct answer will be yes, number B, only enzymes have active site, not receptor. This is not true. Okay, so a receptor is a protein molecule usually found embedded within the plasma membrane surface of the cell that receive the chemical signal from the outside the cell, and when such chemical signal bind to the receptor, they cause some form of cellular and tissue response. Okay, so this is the receptors and these are the signal molecule or ligand. The receptors have an active site where the signal molecule can bind. After the signal molecule can bind the receptor, they share a signal transaction via secondary messenger and cellular response, and the reaction happen inside the cell. Okay, most receptors are membrane bound protein that contain an external binding site for hormones or neurotransmitters. Binding results in an induced fit that changes the receptor conformation. This triggers a series of events that ultimately results in the change in the cellular chemistry. Neurotransmitters and hormones do not undergo a reaction when they bind to the receptor. They depart the binding site unchanged once they have passed their message. The interaction that bind a chemical messenger to the binding site must be strong enough to allow the chemical message to be received but weak enough to allow the messenger to depart. Binding groups are the functional group present in the messenger molecule which are used for binding it to the receptor binding site. Binding regions are the region of the receptor binding site which contain functional group capable of forming intermolecular bonds with the binding groups of the messenger molecule. Okay. So this is a brief introduction about the receptors. So number B, which says only receptors have active site, or only enzyme have active site, not receptor, it is not true because receptors also have an active site where the ligand or messengers can bind. Okay. Now problem number 16, it says which of the following agents activate a receptors to produce a submaximal effect but antagonizes the action of a full agonist. Your options are number A agonist, number B antagonist, number C partial agonist and number D inverse agonist. Which of the following agent activates a receptor to produce a submaximal effect but antagonizes the action of a full agonist? Your options are number A agonist, number B antagonist, number C partial agonist, number D inverse agonist. Again, I will wait for a minute, then I will check the chat box. Okay. Which of the following agent activates the receptor to produce a submaximal effect? What will be the correct answer? Let me know A, B, C or D which you think is the correct answer for problem number 16. Which of the following agents activate the receptor to produce a submaximal effect but antagonizes the action of a full agonist? Number A agonist, number B antagonist, number C partial agonist and number D inverse agonist. Let me check if there is any answer in the chat box for problem number 16. Okay, number C, number C, number C, number C, number C. Okay, B and D, your enzymes, but how can the reaction takes place in enzyme? Enzymes, no, no, no. Okay, actually, the reaction takes place in the active site of the enzyme. Okay, so enzymes are not taking participating in the particular reaction. The enzyme it has the active site. Okay, it has a pocket. In that pocket, the substrate is binding, and reaction is taking place in the particular pocket of the enzyme. It's not like enzyme itself is participating in that particular reaction, but it is providing a surface for that particular substrate to do the reaction. Okay. I think you are confused between this thing. Okay, I have got number C as the answer. I think number C and also number B and D. Okay, okay, good. So for number 16, I have got number C as the answers. Let me check if number C is the correct answer or not. So the correct answer will be yes, very good. It is number C, partial agonist. Okay. So the following terms are used to describe the drug receptor interaction. Agonist, an agent that activates the receptor to produce an effect similar to that of the physiological signal molecule. Inverse agonist, an agent that activates the receptor to produce an effect opposite direction of that of the agonist. Antagonist, an agent that provides an action of an agonist to the receptor or the subsequent response but does not have any effect of its own. Partial agonist, an agent that activates the receptor to produce sub-maximal effect but antagonizes the action of a full agonist. Or ligand is the molecule that attaches selectively to particular receptor or site. Okay, the term indicates affinity or ability to bind without a regard of the functional group. Okay, the agonist and competitive antagonist can be both ligands of the same receptor. So this is a receptor. Receptors and enzyme, there are some distinct difference between receptors and enzyme. Okay, reaction happens when a particular substrate bind to the active site of a enzyme, but no reaction happens when a ligand or a drug binds to the receptor. Only it produces some effect or some physiological effects. Okay, so this can be an agonist, partial agonist, or inverse agonist, or antagonist. So if this in the y-axis we have a effect and this is the log dose, so this is the consecutive activity. Okay, so full agonist produces a full effect. Okay, or the most desired effect, which is effect get by the full agonist. The partial agonist, it produces some effect but not as much as the full agonist. It produces some sub-maximal effect. Antagonist, it does not provide any response. Okay, it does not have any effect of its own. It does not provide any response. Inverse agonist, it produces effect but the effect will be complete opposite to a full agonist. So this graph actually shows very well how this particular things happen. Okay. So yes, the correct answer was partial agonist. Now we will go to the next problem. Problem number 17 says, which of the following enzyme catalyzes the biosynthesis of acetylcholine? Your options, number A, acetylcholine esterase, number B, acetylcholine synthase, number B, choline acyl transferase, and number D, choline acyl, number C, choline acyl esterase, and number D, choline acyl transferase. Which of the following enzyme catalyzes the biosynthesis of the acetylcholine? Again, I will wait for a minute, then I will check the chat box. Okay. Meanwhile, let me know. So in today's class, actually we have learned a lot of things with, apart from the mathematical questions which are lengthy, the answers are, of few questions are lengthy. Okay. You have to read the things properly once I share the slide, and then if you do not understand anything, I am telling it repeatedly, please drop an email before the next class so that I can prepare it accordingly and I can, discuss it in detail in the upcoming classes. Okay. Some things that maybe seem a little difficult, but once you read it by yourself, I think it will be much easier for you to understand these things. Okay. Which of the following enzyme catalyzes the biosynthesis of acetylcholine? Let me know. After this question, I will share my email ID and I will pin it so that you can, write it down before the class ends. Okay. What will be the correct answer? Number A, acetylcholine esterase. Number B, acetylcholine synthase. Number C, choline acyl transferase. And number D, choline acyl transfer. Number C is choline acyl esterase. Number D is choline acyl transferase. Which enzyme catalyzes the biosynthesis of acetylcholine? Let me check the chat box. Let me see if there is any answer for our problem number 17. If there is any answers. Okay. Number B, number D, number D, number D, number D. Number A. Okay, I think I have got number A, number D, and number B also as the answer. Before I show you the answer, let me drop my email ID so that you can note it down. So this is my email ID. Okay, I will pin it. So you can note it down. This email ID. If you have any doubt or any query regarding this class or regarding any topic, you can drop an email before the next class so that I can discuss it in detail. Okay. Now let me show you what will be the correct answer for problem number 17. Yeah. The correct answer will be yes, number D, choline acyl transferase is the enzyme which catalyzes the biosynthesis of acetylcholine. So let us first see the reaction. So this is the acetyl CoA and this is the choline. Okay, and this acetyl CoA and choline both are interact with each other to form the acetylcholine by the enzyme choline acyl transferase. Okay. So this is the cholinergic signaling system. Okay, now first let us understand what is happening here. Okay, so this shows the synapse between two neurons, and these two neurons, the events are involved when the message is transmitted from one neuron to another neuron. Okay, so the first stage involved the biosynthesis of acetylcholine. Okay, so this is the biosynthesis of acetylcholine, acetyl-CoA and choline. They together come together and form the acetylcholine. Okay, by the enzyme this choline acyl transferase. Okay, acetylcholine is incorporated to the. This is the acetylcholine. Let's say, as you can see, this is the acetylcholine. This is inside a vesicle. Okay, acetylcholine is incorporated into a membrane-bound vesicle by the means of the specific transport protein. So first there is a synthesis of acetylcholine, then the acetylcholine goes inside the vesicle, and then arrival of the nerve signal leads to the opening. This is the leads to the opening of the calcium ion channel and an increase of the intercellular calcium concentration. This induces the vesicle to fuse with the this fuse with this position. Okay, now with the cell membrane and release this neurotransmitter or release this particular acetylcholine into the synaptic gap. This is the synaptic gap. Okay, this acetylcholine crosses the synaptic gap now, and then it goes to the binds to the cholinergic receptor. This is the cholinergic receptor. This blue one is the cholinergic receptor in the particular prosynaptic nerve. Okay, and signaling resulting the stimulation of the second neuron. Acetylcholine moved to an enzyme called acetylcholine esterase, which is situated in the postsynaptic neuron. So here we also have an enzyme which is the acetylcholine esterase, which catalyzes this acetylcholine. Now, after doing this, after passing out the signal, where does this acetylcholine go? This acetylcholine goes to the another receptor, which is known as another enzyme, which is known as the acetylcholine esterase. Okay, this acetylcholine esterase hydrolyzes the acetylcholine, and it basically it recycles this component, the acetylcholine, for this choline and acetic acid toward this presynaptic nerve. Choline is taken up by this presynaptic nerve to transport to the another cycle, and this okay, so this is the cycle, this is the cholinergic signaling system, how it works. Okay, there all the things are shown in the picture and all the steps are written here. Just read it by yourself one more time. If you have any query, let me know. The cholinergic nervous system involves nerves which use the neurotransmitter acetylcholine as a chemical messenger. This include the motor nerve which innervate a skeletal muscle, nerves which synapse with other nerves in the peripheral nervous system, and the parasympathetic nerve nerves innervating cardiac and smooth muscle. There are two types of cholinergic receptor: muscarinic receptor, which are present in the smooth and cardiac muscle, and nicotinic receptor, which are present in the skeletal muscle and in the synapses between the neuron. Acetylcholine is hydrolyzed by the enzyme acetylcholine esterase when it deposits the cholinergic receptor. The hydrolytic product choline is taken up by the presynaptic neuron and acylated back to the acetylcholine. The cholinergic receptor and the enzyme acetylcholine esterase are useful drug targets. Acetylcholine cannot be used as a drug because it is rapidly hydrolyzed by the acid and enzyme. It shows no selectivity for different types and subtypes of cholinergic receptor. Okay. So this is all about the cholinergic signaling system, which is very important. Okay, and acetylcholine is the very crucial, one of the very crucial neurotransmitters. We will read about the neurotransmitters again in our. I think you have the neurotransmitters in your week 7 or 8. Okay, then also we will discuss about these neurotransmitters in detail. Now let me go to the next problem, problem number 18, which says which of the following is not an example of a G protein coupled receptor. Your options are number A muscarinic receptor, number B adrenergic receptor, number C kinase gated receptor, and number D opioid receptor. Which of the following is not an example of G protein coupled receptor? Muscarinic receptor, adrenergic receptor, kinase gated receptor, or opioid receptor? Again, I will wait for a minute and then I will check the chat box. Let me know A, B, C or D, which of the following you think is the correct answer for problem number 18. Which of the following is not an example for G protein coupled receptors? Muscarinic receptor, adrenergic receptors, kinase gated receptor, or opioid receptor? Which of the following is not an example of G protein coupled receptors? Let me check if there is any answer in the chat box. Okay, I got many answer. Number C, number A, number C, number C. Majority is toward number C and one I have received number A as the answer. Let me check which will be the correct answer. So, the correct answer will be yes, number C, kinase gated receptor. It is not an example of G protein coupled receptor. So, G protein coupled receptors are some sort of the most important drug targets in medicinal chemistry. Okay. Let me just highlight few point. So this is the cell membrane. Here is the receptor, which is the and there is a binding site, and this there is a protein called the G protein. When the binding site is closed, then the binding when the binding in the presence of the messenger, the binding site is get opened, and this G protein bind to the binding site. Okay, and the particular signal transaction happen. And so this is the activation of the G protein coupled receptor and G protein. The G protein coupled receptor is some sort of most important drug target in medicinal chemistry. The around 30% of the drugs in the market act by binding to these receptors. Okay, so this which makes it one of the most crucial drug targets in medicinal chemistry. They are activated by hormones and slow acting neurotransmitter. They include the muscarinic receptor. Okay, muscarinic receptors, adrenergic receptor, and opioid receptor. So these three are the examples of the G protein coupled receptors. The response from activated G protein coupled receptors are measured in second. The slower than the response for the ion channel and faster than the response. Their result are slower than the response from the ion channel and faster than the kinase ligand gated receptor, which takes a matter of minutes. There are a large number of different G protein coupled receptors interacting with important neurotransmitters such as acetylcholine, dopamine, histamine, serotonin, glutamate, and noradrenaline. Other G protein coupled receptors are activated by peptide or protein hormones such as the enkephalins and endorphins. G protein coupled receptors are membrane bound protein that are responsible for activating the protein called the G proteins. Okay, these later proteins act as a signal proteins because they are capable of activating or deactivating the membrane bound enzymes. Consequently, activation of the receptor by a chemical messenger influences the reaction that takes place within the cell. The receptor protein is embedded within the membrane and the binding site for the chemical messenger exposed on the outer surface. On the inner surface, there are another binding site which is normally closed. When the chemical messenger binds to its binding site, the receptor protein changes its shapes and opens the binding site for the inner surface. This new binding site is recognized by the G protein which then binds. Okay, so this is shown in this frame 1, 2 and 3. The G protein attached to the inner surface of the cell membrane made up of the 3 protein subunit, and the once binds to the receptor complex destabilize and fragments to a monomer and dimer. This interaction with the membrane bound enzyme continue to the single transduction process. The several different G proteins which are recognized by different type of receptor. Some are activated by subunit. These G proteins are inhibitory effect for the membrane bound enzyme, while others have a stimulatory effect. Nevertheless, the mechanism by which G protein activated by fragment is the same. There are substantial amplification of the signal in this process. Once activated receptor activates several G proteins. Okay, so this is all about the G protein coupled receptors. So now I will go to the next problem. So the problem number 19 says how many subunits are present in the ion channel structure. Your options are number A 5, number B 4, number C 7 and number D 9. How many subunits are present in ion channel structure? 5, 4, 7 or 9? Let me know A, B, C or D, which of the following you think is the correct answer for problem number 19. How many subunits are present in the ion channel structure? Since we do not have much time, let me just discuss these answers. Okay. So in the number of subunits present in the ion channel structure will be 5. Okay. So generally ion channels are another receptor. So different receptors that are important for your syllabus is the G protein coupled receptors, also this ion channel receptors and kinase gated receptors. Okay. So some neurotransmitter operate by controlling the ion channels. What are these ion channels and why they are necessary? Okay. So the membrane is made up of the bilayer of the phospholipid molecules. So the middle of the cell membrane is fatty and hydrophobic. Okay. Such barrier makes it difficult for the polar molecule or ions to move in or out of the cell. Yet it is important to these species to cross. For example, movement of the sodium and potassium ion across the membrane is crucial for the function of our nerves. It seems an intracerebral problem. This seems an intracerebral problem. Once again, the ubiquitous proteins provide an answer by forming the ion channels. Okay. So ion channels are the complexes made up of the 5 protein subunits. This is important to remember. 5 protein subunits are present in the ion channel which traverse the cell membrane like this. By the the center of the complex is hollow and lined with polar amino acid, making it hydrophilic and tunnel or pore. Ions can pass through the fatty barrier of the cell membrane by moving through these hydrophilic channels or tunnels, but there has to be some control. In other words, there has to be some lock gate that can be opened or closed as required. This makes sense. This lock gate should be controlled by receptor protein sensitive to the external chemical messenger, and that is exactly what happens. In fact, receptor proteins are the integral part of the ion channel complex and one or more constituent protein subunits. The resting state, the ion channel is closed, like the lock gate is shut. However, when a chemical messenger binds to the external binding site, it causes the induced fit, which causes the protein channel to change shape. This in turn causes the overall protein complex to opening up the lock gate and allowing the ion to pass. Okay, the operation of the ion channel explains why relatively small membrane neurotransmitter molecules released by the neuron are able to have such significant biological effect to the target cell. By opening few ion channels, several thousand ions are mobilized for each neurotransmitter molecule involved. Moreover, the binding of the neurotransmitter to an ion channel results in the rapid response. measured in matter of millisecond. That is why synaptic transmission of the signal between neurons usually involve the ion channels. Ion channels are specified for certain ion. For example, different cationic ions for sodium, potassium, calcium ion channels are there, and for also anionic ion channels for chloride. The ion selectivity for the different ion channel is dependent on the amino acid lining of the ion channel. It is interesting to note that the mutation of just one amino acid in the area is sufficient to change the cationic selective ion to anionic selective ion channel. Okay, so this is the structure. 5 subunits are there: alpha, gamma, alpha, gamma, and beta. Okay, so 2 to alpha, one beta, one beta, one delta, and one gamma is there. So this is the pentameric structure of the ion channel. This is the traverse view. Okay, and this is the number B is a traverse view of one including neurotransmitter region. Okay. So this is the neurotransmitter regions inside any every alpha, gamma, and beta, delta. 5 protein subunit make the ion channel are glycoprotein. This is important to remember. Okay, but we will refer them as normal only protein. The protein subunit are not identical. The ion channel of the nicotinic cholinergic receptor made up of the subunit of 2 alpha, beta, gamma, delta. Ion channel controlled by glycinic receptor made up of the 5 subunit of 2 different types, 3 of alpha and 2 of beta. Okay, the receptor protein on the ion channel controlled by glycine is the alpha subunit. So this is not you can read it by yourself. It's not much important. It's more specified. So remember, 5 protein subunit make the ion channels. Ion channels are the tunnels which have the hydrophilic amino acid lined inside each other, which will which enables the ions like sodium, potassium, chloride to pass through the cell, the hydrophilic cell membrane. But it is not like the ion channels will be open all the time. Only when a particular neurotransmitter binds to the receptor and send the signal, only then this ion channels will open and it will pass the certain ions. Okay, and one mutation in this particular ion channel will can make a particular cation ion channel to an anion ion channel. Okay, this is can also be possible. Short notes can be given to you regarding this ion channel and all these things. You can read this slide particular properly and then you can make a short note by yourself. Okay, but the figures are very important. If you have to write a short note on ion channel, it is expected that you will give this figure at least. Okay. Now the last question, it says ATP is required as a cofactor to provide the necessary phosphate group, and the active site remains long open as long as the messenger molecule is bound to dash receptor. Your options were number A G protein receptor, number B ion channel receptor, number C kinase receptor, and number E ligand mediated receptor. And the correct answer will be kinase link receptor. Okay, ATP is required as a cofactor in case of this kinase linked receptor. So as you can see, these are the receptor tyrosine kinase proteins which are the inactive acts as an inactive monomer. Here we have this tyrosine. This is the signal binding site. When the signal molecule binds to this position, this dimer is formed. Okay, but this activated tyrosine when this dimer is formed, this get activated. ATP is converted to ADP, and this particular receptors are fully activated by this phosphorylation, which is happen with help of the ATP, and then this cellular response and signal transduction happen. So this is basically that's how the kinase link receptors are work. Okay, so kinase, remember, kinase is an enzyme which catalyzes the conversion of the ATP to ADP, and the phosphorylation happens in the substrate. Okay, when in the receptor this phosphorylation happens, these receptors are known as the kinase link receptors. Okay. So this was the last question of today's session. So today we majorly discussed about the various enzyme catalyzed reaction. We learned various about many mathematical question we solved, and also we learned about different kind of receptors. Okay, so this was the week 2 also. Okay, problem number 18. What was it? So problem number 18, it's basically about the G protein couple receptor. Okay, so G protein couple receptor, this is also one kind of receptors which once activated, it binds with the G protein. Okay, and this G proteins are act as a signal protein because they are capable of activating the membrane bound enzyme. I suggest you go through this slide once since the time is also up for today. One thing I will suggest, Sujata, please go through this slide. Okay, once I share, please go through the slide. If the problem persists regarding the G protein couple receptor, let me know. I will repeat it in the next class. Okay, will it be fine? because this is a very long topic. I hope when you will read the thing, I think it is better you will understand. Okay. If you do not understand this topic, let me know before starting the next session. At first, I will repeat this topic and then I will go through the upcoming class topics. Okay. I think it will be better. Okay, so that's what I am saying, that we have discussed a lot of things in today's class. And things some things are very new, especially the receptor things are new and a little bit complicated, it might seem. Once please go through the slides, and if you find that you are not understanding a particular topic or a particular question, just drop an email before the next class. I will then repeat it in the upcoming class before starting the week 3 course content. Okay. So thank you all for joining. I will close the session for today. This presentation and this class recording, I will be uploading to the NPTEL slide, which you can check by tonight or tomorrow morning. Okay. Thank you all. Thank you for joining. I will closing the session for today. Thank you.